Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, so the human brain, it develops from like this tiny cluster of cells, right? And becomes, well, basically the command center for like everything. It's incredible. And sometimes even the tiniest glitch in our genes can throw things off, especially with things like epilepsy.
It really is amazing when you think about it. I mean, we don't often stop to think about all the crazy, complex things that go on just to make our brains work. It's really only when something goes wrong that you realize just how delicate this whole process is.
Exactly. And that's what we're looking at today. We're focusing on this family of genes called cyclin-dependent kinase-like.
CDKL.
Yeah, CDKL, and their role in some of these neurodevelopmental disorders. You might have heard of CDKL5. It's linked to a rare genetic disorder that causes seizures and developmental delays. But there's this new research suggesting that other members of this gene family, like CDKL1 and CDKL2, well, they might be involved in similar things.
Yeah, this is really interesting. The CDKL gene family has been studied a lot, but this new research is kind of shining a light on some of those lesser-known members. It could help us understand a lot more about how these genes affect brain development and how they work overall.
So we've got this brand new scientific paper straight from the researchers at Baylor College of Medicine, and we've got you, of course, to help break it all down. Ready to jump in?
Oh, absolutely. Let's do it.
So it seems like the scientists over at Baylor, they were really curious about these two genes, CDKL1 and CDKL2. They wanted to know if maybe they were contributing to these neurodevelopmental issues in a similar way to CDKL5.
Yeah, that was definitely their starting point. They began by analyzing genetic data from individuals who were showing signs of those neurodevelopmental problems, ones that resembled what we see in people with CDKL5 deficiency disorder.
And did they find anything interesting?
Oh, they did. They found 4 brand new variations in CDKL2 in 5 different individuals. And on top of that, they found 2 new variations in CDKL1 in those databases that have, all the information from people with similar developmental challenges. Essentially, they're just slight changes in the DNA sequence of these genes.
So, okay, they found these variations. That's pretty cool. But how did they figure out what those genes were doing and if those variations were actually the cause of the problems?
Well, this is where things get really fascinating, actually. They used, fruit flies.
Fruit flies? Wait, how can studying flies tell us anything about human brains?
I know it sounds strange, but it makes sense. Fruit flies, they actually share a lot of genes with humans, including one very important gene called CDKL, and it's surprisingly similar to all 5 of those human CDKL genes.
So by studying this one CDKL gene in flies, they can learn about the whole CDKL family in humans. Kind of like using a simpler model to understand something much more complex.
Right. Yeah, exactly. It's like, imagine you have a blueprint that shows the common features of, different types of cars. So in this case, fruit flies, they're the blueprint. They give us a really powerful way to investigate what all these genes are doing.
Okay, I'm with you. So what exactly did they do to these flies? How did they mess with this CDKL gene?
Well, they did a few things. First, they got rid of the CDKL gene in the flies completely to see what would happen. And, it was pretty dramatic. 90% of the flies died.
90%. So this isn't just some random gene. It's super important for survival. What about the flies that survived? Were they normal?
They weren't completely normal, no. The surviving flies had a bunch of problems. They had trouble moving, lost their hearing, and even started having seizures when they got too hot. It showed just how important this gene is, not just for survival, but also for the brain to develop normally and work right.
Okay, so deleting the gene was bad news. But I bet they then tried to fix things, right? Did they put the human versions of those genes into the flies?
You got it. They put those normal human CDKL1, CDKL2, and CDKL5 genes into the flies that were missing their own CDKL gene. And get this, it actually fixed a lot of the problems. The flies could move normally again, their hearing came back, and the seizures stopped. It just shows how similar the fly gene and the human genes are.
It really is amazing, like you said. These flies, they become these little living test tubes. It lets us change genes around and see what happens in a way that you just couldn't do with people.
And the results are super clear, too. But what happened when they took those human gene variations, the ones they found in people with those neurodevelopmental problems, and put those into the flies? Did that fix things, too?
Well, that's where things got a little more complicated, actually. When they used those variations, things didn't go back to normal completely. The flies still had some problems.
So it sounds like those variations aren't acting like the regular human genes. Does that mean those variations are actually causing those problems in humans? That's exactly what the researchers think. It seems like those variations aren't just, sitting there doing nothing. They might actually be messing with how the normal genes are supposed to work. They call this a dominant negative effect.
Dominant negative. So those variations are kind of going rogue and messing things up for everyone.
That's a good way to put it. Imagine you've got a team working on something, and one person not only isn't helping, they're actively making things worse for everyone else. That's kind of what's going on here, but on a tiny molecular level, even though there's a normal copy of the gene, the messed up one is throwing everything off balance.
Wow, so even one bad copy can cause so much trouble. It's kind of like a bad apple spoiling the whole bunch, And this makes me wonder, what does all this mean for actually treating these conditions?
This is where the research gets really exciting. If we can figure out exactly what these genes are doing and how those variations mess things up, we could start developing treatments that target those specific problems.
So instead of just treating the symptoms, we could fix the actual root of the problem. That sounds pretty amazing. But I'm guessing it can't be easy to turn these findings into actual treatments for people.
It definitely has challenges. I mean, fruit flies are great, but they're not people, obviously. To really get to treatments, we need more research, especially in animals that are more like us, like mice.
So the next step would be to confirm all of this in mice. It seems like we're slowly piecing together this huge puzzle, and each study adds another piece to the picture. What other implications does this research have?
It's not just about these specific genes. This study shows just how important it is to understand the connection between genes and brain development. It's like we're learning that even small changes in our genes can have huge effects on our brains, on all those complex networks that make us who we are.
It really makes you realize how much we still have to learn about the brain and how genes influence it all. So what could researchers do to actually develop treatments based on this? Would it be medications, gene therapy, or something completely different?
There are a few paths we could explore. One option is to develop drugs that specifically target the messed up proteins created by those variations. Imagine a tiny off switch that shuts those troublemakers down without affecting the other genes.
So basically silencing the bad guys without messing with the good guys. I like it.
Another possibility, although it's a bit more futuristic, is gene therapy. We could either give people a working copy of the gene or even try to fix the variations themselves using gene editing, like that CRISPR-Cas9 technology you might have heard of.
CRISPR, yeah, that's the one where they can basically edit genes like they're using tiny scissors, right?
Exactly. It's pretty revolutionary, but it's still early days for CRISPR.
Even if those treatments are years away, it's still incredible to think we might actually be able to fix these conditions at the genetic level. But what are some of the challenges, especially when you're talking about something as complex as the brain?
Well, one of the biggest things is making sure any treatment is both effective and safe. The brain is so complicated, and any changes we make could have unintended consequences. We have to be super careful with testing and clinical trials to make absolutely sure we're not causing more harm than good.
Yeah, it's like walking a tightrope. You're trying to fix something without messing up the entire balance of the brain.
Right, exactly. It means we need to understand not just those specific genes, but also how they interact with all the other stuff going on in the brain.
It sounds like this is just the beginning. It's a whole new area of research, not just for these specific conditions, but for how we think about neurodevelopmental disorders in general.
I think so. This study is a perfect example of how even the most basic research can lead to some really huge breakthroughs. By figuring out what these genes are doing, the researchers have opened the door for future studies to come along and take it even further.
It reminds us that science is all about being curious, and that research really has the power to change people's lives. But before we get carried away, were there any limitations to this study that we should keep in mind?
They mentioned that they only looked at a small group of people with these variations. To really understand how common these variations are and what kind of impact they have, we'll need more research with more people from different backgrounds.
So it's about expanding the research to see if these findings hold up in a bigger, more diverse population. That makes sense. You want to make sure this isn't just a fluke.
Exactly. And remember, as great as fruit flies are, they aren't humans. We need to confirm everything in mammals that are more like us, like mice.
So basically, we need to build on this research and make sure it applies to people. It's a good reminder that science is always a work in progress. It's not about getting to some final answer.
Well said. It's a constant process of exploring and refining our understanding of the world. And in this case, that exploration has given us some amazing insights into how the human brain works. It's amazing how much we've learned from those tiny flies, you know? Who would have thought they could tell us so much about the human brain?
It's a good reminder that even the simplest creatures can hold important clues about how we work. And in this case, they've opened up some pretty amazing possibilities for treating these neurodevelopmental disorders.
Absolutely. We've talked about developing new drugs and even using gene editing with CRISPR, but how long do you think it will actually be before these treatments are available?
Yeah, that's the big question, isn't it? It all sounds so promising, but how realistic is it to think we'll see these treatments soon?
Well, we have to be patient. Developing new treatments, especially when you're dealing with gene editing and the brain, it takes time. You have to do tons of testing, make sure it's safe, go through clinical trials, and get approval before anyone can actually use it.
So we're not going to see these treatments overnight.
Right. It's more like a marathon than a sprint. But even though it's a long road, I think the progress we've made in the last few years with both new drugs and gene editing, it's really encouraging. Things are moving faster than ever, and I'm optimistic that we'll see some real breakthroughs soon.
It's pretty amazing to think about what these treatments could mean for people with these conditions. But with all this excitement, we also need to think about the ethical side of things, especially when it comes to gene editing.
Oh, absolutely. Gene editing is powerful, and it could change medicine completely, but it also brings up some serious ethical questions that we all need to talk about. We need to have open and honest conversations about the benefits, the risks, and where we draw the line with gene editing to make sure we're using it responsibly.
Yeah, we don't want to just jump in without thinking about the consequences.
Exactly. It's all about finding that balance, pushing the boundaries of science, but doing it in a way that's ethical and responsible.
Well, as we wrap up our deep dive into these CDKL genes and neurodevelopmental disorders, I'm just struck by how complex the brain really is and how much those genes influence its development.
It really is mind-boggling, isn't it? And this research just shows how even the smallest changes in our genes can have huge effects on our health and well-being. It highlights just how important it is to keep researching and to keep trying to unravel those mysteries of the brain and to develop treatments for people who are struggling.
It reminds us how much we still have to learn, but with every new discovery, we're getting closer to understanding ourselves and how life works.
And I think, most importantly, this research gives us hope. We've come so far in understanding these complicated genetic disorders, and there's so much potential to develop treatments that could really make a difference for people.
It shows what we can achieve when scientists are curious and dedicated to solving these problems.
And to everyone listening, if you're interested in genetics and neuroscience, keep learning. There's so much information out there. You can find stuff online, at the library, and through all kinds of educational programs.
And remember, we all have a part to play in supporting research and advocating for policies that help science move forward. By staying informed, talking about these issues, and supporting scientific work, we can create a future where these disorders are a thing of the past.
That's a great point. Science is a team effort, and we can all contribute to unlocking the secrets of the brain and creating a healthier future.
And on that note, we'll wrap things up here. Thanks for joining us on this incredible journey into the world of CDKL genes and neurodevelopmental disorders. Until next time, keep exploring the wonders of science and the amazing potential of human ingenuity.
Thanks for tuning in to the Health Headliner podcast by Todo Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.